Welcome to the next lesson on master regulatory genes. In this lesson, we're going to be looking at how master genes across a number of animal phyla demonstrated specifically by the expression of BMP4 in beak formation of Galapagos finches and jaw formation of cichlid fishes in Africa result in a number of phenotypes. So master regulatory genes, they control the expression of regulatory genes. Specifically, an example that we've already learned is the lacoperon, where the lacoperon is regulated by the lactose sugar, and the lactose sugar will regulate transcription of lactase. So a master regulatory gene that controls the expression of structural genes during embryonic development. So master regulatory genes will control the rate, the timing, and spatial expression, so the location in which that gene may be expressed. An example of such is, here is gene X, a master regulatory gene. It will release a transcription factor which will bind to a number of other genes, and that will turn on, as in gene A, B, C, and E, or will turn off, as in gene D, that particular gene. If that gene is turned on, it can result in a number of different phenotypes. Take this picture over here. When we turn on this particular gene via a master regulatory gene, we have development of the forehead or elongated in here. When we have this particular yellow gene turned on, we develop various different structures in the tail of that organism. This happens during embryonic development. So cichlid fish are the specific example that we're going to be looking at in terms of regulatory genes, as well as Galapagos finches. So first of all, cichlid fish are a fish in Africa, and there's over 500 species. So the emergence of these vast amount of species is evidence for adaptive radiation. That's a type of divergent evolution. So that means they all come from one common ancestor, and they share similar traits, but have diverged to become different species, and that's due to different selection pressures. So you can see here in the diagram on the right, there are various different locations in which those fish exist, all different lakes, and in those lakes there are different selection pressures, which has resulted in a vast array of particular species that are present in that species, known as adaptive radiation. So specifically, with cichlids, they have expression of the master regulatory gene, BMP4. So they have differences in their jaw structure, which is a result of the regulatory gene being expressed. So you can see here that when we have different levels of BMP4 being expressed, we have differences in jaw. Specifically, when we have low-level BMP4 expression, we have longer jaws with spaced teeth, just like in option C here, or picture C. When we have high-level BMP4 expression, we have short, robust jaws, and they obviously will have different feeding environments, so they'll eat different things. When we have intermediate or medium expression of BMP4, we have a jaw structure which looks like this. Let's take a look at Galapagos finches. Again, they have a vast difference in their beak shape, and a number of different species have resulted. This is due to adaptive radiation. So here there are a number of islands which Galapagos finches have inherited or do inhabit, and those different islands will have different selection pressures, which will have resulted in a number of different phenotypes. In terms of BMP4, the master regulatory gene, and CAM, which is calmodulin, a particular gene involved in the formation of beaks in the embryo, will change the shape of the beaks. If we have a high BMP4 expression, we're going to have, and a low CAM expression, we'll have a shortened beak here. It's a very fat beak, but it's quite short. When we have moderate BMP4, we have a longer beak, and it's much thinner. Again, a moderate BMP4, but higher CAM expression is an elongated beak, and it's much shorter in its depth here. And we have high CAM, a low BMP4, we have an elongated beak. This tells you that BMP4 is responsible for the thickness of the beak, and CAM is responsible for the length of the beak. So you need to be able to understand that regulatory genes are responsible for changing the beak shape and size in Galapagos finches and jaw formation in cichlid fish. This is the summary of the main ideas: adaptive radiation, just refreshing that term, and master regulatory genes and how this alters phenotypes. These are the key terms, and these are the points that you need to be able to measure your level of understanding against. So you should be able to explain adaptive radiation and explain how master regulatory genes control or contribute to fast differences in phenotypes, with specific example of BMP4 and CAM, and the examples of those two are cichlid fish and Galapagos finches. Thanks for watching.